There's a new warning from a group of over 24 biologists. They are calling for a ban on mirror cell research, warning it's too dangerous. Mirror bacteria is constructed from mirror images of molecules found in nature. They say it could put humans, animals, and plants at risk of lethal infections and could lead to the collapse of entire ecosystems. In a 300-page report published by Stanford University, researchers say mirror bacteria could not evolve from existing life, but their creation will become increasingly feasible as science advances. Their altered interactions with natural organisms could lead to profound and highly consequential effects of those natural organisms. Now, our next guest is one of those scientists raising those concerns. Dr. Michael Kay is a professor of biochemistry at the University of Utah who builds mirror molecules that could be used to develop therapeutics. We reached him in Salt Lake City, Utah. Thank you, sir, for taking the time.
Thank you very much.
Okay, very quickly and as simply as possible, if you could, let's just start with perhaps explaining a little bit, very simply, if you could, what exactly mirror molecules are.
Sure. So all of the natural biomolecules, things like DNA, proteins, and sugars, have a certain handedness to them, kind of like your hands. It's theoretically possible to make versions that are the mirror image, like your opposite hand, of those molecules. But those types of molecules can only be made synthetically. They don't occur naturally. And these are molecules that have a lot of potential, especially for therapeutics, because they're not recognized and degraded by the body, and they have the potential to be very long-lasting therapeutics.
So potentially some good could come out of this, but we know there are many concerns that scientists have about this kind of organism. You once yourself, we know, had aspirations to create mirror life, but now you don't. So talk me through why.
So the reasons are partly because of this, group's efforts to research and consider what the potential risks are of mirror life. And we've come to the conclusion, based on what we know so far, that those risks are very serious and that the benefits are very modest in comparison. And it turns out that you can access all of the benefits of mirror image molecules and therapeutics using chemical techniques. And so the key distinction here is making a self-replicating mirror entire organism like a bacteria is where the risk lies, because such an organism could grow unchecked.
And there is, if I understand this correctly, already a mirror molecule in science. So what's the difference between those molecules and mirror life?
So mirror life has the critical property of being able to self-replicate. It's something that can take in food from the environment and grow like natural life, and therefore compete with natural life. Mirror molecules are chemically synthesized and inert. Once they're made, they cannot do anything else in terms of they can't reproduce themselves, they can't grow, they're just inert compounds. And so they're in wide use already in humans and in science and have already been proven to be very safe and effective. And so there's really no risk there. The risk only comes into play once you create an entire organism of mirror image components that can grow, take in food from the environment, and potentially compete in a hostile way with natural life.
Talk to us a little bit more. What is that risk? What is the big concern?
The big concern is that using bacteria as the key example, bacteria are kept in check from growing out of control in the natural environment by many different defense mechanisms. Our immune system is a great check against their growth in causing infections in humans. There are many predators of these bacteria that help to keep their population in check. And many other organisms eat these organisms and help to keep them in check. And so it is likely, based on our current information, that a mirror organism would be able to avoid all of those natural defense mechanisms. And therefore, if it was able to find a good source of food, would be able to grow unchecked in the environment. And if it were to get into humans or plants or animals or the ecosystem, it's something that would be, not controlled by any of the natural control mechanisms that otherwise exist.
So Dr. Kay, how close are we actually to creating mirror cells?
We're not that close. And so our group really determined that based on the very fast paced advances in underlying technologies behind chemical synthesis and synthetic biology, that it is now seeming apparent that within our lifetimes, we estimate in the next 1 to 3 decades or so, that this could become feasible if there was a highly concerted effort, an international consortium, major funding, from government agencies and others, that this could become a reality in that kind of time frame. But it is not feasible today, and really not that close to being feasible today. However, we thought that given the rapid advancement of technology, that this is now looking plausible enough in the not too distant future that this is the time to have a reasoned discussion with a wide range of stakeholders across the world to really think about how we can head off the significant risks of ultimately making mirror life.
All right. Well, we thank you so much for your time today and explaining it all to us. Very much appreciated. Michael Kay is a professor of biochemistry at the University of Utah.